Cells serve as the fundamental building blocks of all living organisms, forming the structural and functional foundation of life. According to cell theory, every organism is composed of one or more cells, and all physiological activities depend on the collective function of these cells. In multicellular organisms, specialized cells work together to maintain homeostasis, with different types of cells performing distinct roles. For example, muscle cells contract to enable movement. Neurons transmit electrical signals for communication, and red blood cells transport oxygen throughout the body. This specialization reinforces the principle of complementarity, which states that a cell's function is determined by its internal structures. The presence of organelles such as mitochondria, ribosomes, and the nucleus allows cells to carry out specific tasks essential for life. Furthermore, the continuity of life is ensured through cellular reproduction and division. Somatic cells divide through mitosis to support growth and repair, while gametes undergo meiosis to maintain genetic diversity in sexually reproducing organisms. The cytoplasm, which fills the space between the plasma membrane and the nucleus, plays a vital role in cellular function. It consists of cytosol, organelles, and various inclusions. Cytosol, the fluid component of the cytoplasm, contains dissolved nutrients, ions, and proteins, providing the medium necessary for biochemical reactions. Suspended within the cytosol are cytoplasmic organelles, which are specialized structures that perform essential cellular processes such as energy production, protein synthesis, and waste degradation. The cytoplasm contains inclusions, which are stored materials like glycogen granules in muscle and liver cells, lipid droplets in fat cells, and melanin pigments in skin cells. These inclusions play key roles in energy storage, structural integrity, and cellular protection. Among the most crucial organelles in the cytoplasm are the mitochondria, often referred to as the powerhouse of the cell due to their role in ATP production through aerobic respiration. Mitochondria contain their own DNA, RNA, and ribosomes, allowing them to replicate independently of the nucleus. Their highly folded inner membrane, known as cristae, provide a large surface area for ATP synthesis, which is essential for fueling cellular activities. Another key organelle is the ribosome, responsible for protein synthesis. Ribosomes exist in two forms: free ribosomes, which produce proteins used within the cell, and bound ribosomes, which attach to the rough endoplasmic reticulum and synthesize proteins for secretion or membrane incorporation. The endoplasmic reticulum plays a further role in protein and lipid processing. The rough ER, studded with ribosomes, is heavily involved in protein synthesis and transport, while the smooth ER specializes in lipid synthesis, detoxification of harmful substances such as drugs and alcohol, and calcium storage in muscle cells. Proteins and lipids synthesized in the ER are then processed by the Golgi apparatus, which modifies, sorts, and packages these molecules for transport to their final destinations. This organelle is essential for proper protein folding and trafficking, particularly in cells that produce large amounts of secretory proteins such as glandular cells. The lysosome, another important organelle, contains digestive enzymes that break down waste materials, damaged cellular components, and foreign particles. Lysosomes play a crucial role in cellular cleanup and turnover, ensuring that cells remain functional and free of harmful debris. A failure in lysosomal function can lead to serious disorders such as Tay-Sachs disease, in which impaired lysosomal enzyme activity results in toxic buildup within neurons, leading to neurological deterioration.